This is a set of slides that describe an idea for trying to understand how events among different chromosomes interact with each other. The genomic wall is a tool for understanding genomic behavior. The basic hypothesis is that many diseases have phenotypes that involve genes on different chromosomes, and that diploid genomes utilize genes on both sets of chromosomes. There needs to be a tool that can show the active elements on all chromosomes, and the tool should be independent of any particular theory. The properties of a genomic wall are such that there should be perhaps one display and processor for each chromosome in the human genome, for example, and to provide a support program running on each of these processors to select genomic or theoretical properties as a function of the position in the chromosome, and to provide a language for describing and manipulating the properties in a very general fashion. The displays and processors are now very inexpensive. An Intel processor with 1 gigabyte of memory can now be housed in a little box, 6 inches square by 2 inches deep. And a 5x5 array of these things would allow one to have a display and a processor for each chromosome. So here's a mock-up of how the 25 displays might look. I mean, the 23 chromosomes of a human, and then one could use the other two for the statistics and interchromosomal displays. So something on the order, size doesn't matter. The size could be either the 17-inch displays or smaller or larger. It doesn't make any difference. The mock-up of the back would be that you have a processor associated with each display, and then a switch which would route signals to each of the displays. I've been using Mage and King from the Richardson Laboratory at the Duke University Medical School for the three-dimensional display of data on each chromosome. And as you'll see in the various other YouTube videos that I've made, it's possible to show each chromosome as a spiral, and then the concentric spirals show different properties, and then color is used to differentiate even further. The idea that we have, of course, is that the genomic wall would be able to accept any GMOD or UCSC viewer track, and the tracks developed in GMOD would be able to be imported into the genomic wall. The genomic wall provides support for the connectron theory that I've been developing for the last decade or so. Connectron theory provides for genome-wide information, both of gene coding and non-coding transcription events that control the expression of other transcription events on the same or different chromosomes. The genomic wall presents the requisite level of genomic information because you have these 25 displays. And a simulation program that would run on yet another computer, let's say, would have the data for all of the 23 chromosomes, and you'd be able to let this simulation run and then watch the behavior of the simulation on the 25 displays. Of course, when you go to a diploid genome and its simulation, you might want to have two genomic walls with 46 or 50 displays. I mean, this is something we haven't seen yet. So my view is that the genomic wall is an evolutionary tool made possible by small computer processors and inexpensive displays that would enable scientists to study the regulatory interaction in any genome. Revolutionary theories of whole genome events may emerge from the use of the genomic wall display.